As we have seen in our previous lectures, many of our important biomolecules in the body are actually biopolymers that are formed from small individual molecular monomers. So, for example, in the case of proteins, we looked at the structure of proteins by starting at the structure of our amino acids, and then we saw how those amino acids would be linked together to form our proteins, and our proteins could then combine with other proteins or other ligands or binding partners in order to carry out the various functional roles in the body. In the next few lectures, we're going to be taking a close look at sugars or carbohydrates. Again, just like our proteins, small sugar monomers will be linked together to create larger polysaccharide sugar polymers. These polymers, of course, can have a structural role, such as the cell wall in plant cells, but our sugars will also have a very key role in metabolism, with those sugars acting as a fuel source for our body's use. So what actually is a carbohydrate? Well, the name of our carbohydrates actually gives us a direct hint. So the general formula of our carbohydrates is CnH2ON, indicating that we have, for every carbon in our carbohydrate, we also have a water molecule to go along with it. Hence the term carbohydrate, right? It's essentially carbon that's hydrated with water. Now, of course, many of our carbohydrates are produced from carbon dioxide and water through photosynthesis in plants, which then we can consume those plants in order to take those carbohydrates into our body. But our body is also able to produce its own carbohydrates as well through our various reactions and metabolic processes. Similar to our proteins, there's a wide range of different kinds of carbohydrates. We can, of course, have our small carbohydrates, which will also act as monomers to build our larger carbohydrate polymers, but will have their own roles as well. And then we can have our very large carbohydrate polymers, and these can be some of the largest polymers that we have in the body. For example, amylopectin, which we'll look at later, can have over 200 million grams per mole in molecular weight. Right? So we can have these really huge sugar carbohydrate structures built up. As I mentioned before, there are a wide range of functional roles for our carbohydrates as well. First, energy use and energy storage is one of the most common forms that you might think of, but they also play a key structural role, and they also act as informational molecules to help in passing signals between cells. Our carbohydrate complexity can be increased even further through linking with proteins and lipids. Right? So our biomolecules aren't just separate classes of molecules that don't interact with each other. They can actually be combined together to create even more variety and complexity in our biomolecules. The structure of our carbohydrate monomers tends to be a bit simpler than our protein monomers of our amino acids. As I mentioned before, our basic formula for our carbohydrates is CnH2ON. All of our monomers for our carbohydrates will use this basic formula, although we will see some modified sugars later that will change up this formula a bit. So if we want to name our carbohydrates, we can just use a few simple rules to help us identify the different kinds of carbohydrates that all share the same formula. So our first rule relates to the number of carbon atoms, right? The first thing we need to know is how many carbons we have. And we can use, of course, a prefix to indicate the number of carbon atoms. And then we're going to use an ending, as is typical with our IUPAC nomenclature, in order to indicate that we're dealing with a sugar or a carbohydrate. That ending is this OSE. So a 3-carbon sugar would be a triose. Notice here that we're not using our typical IUPAC prefixes, but rather we're using kind of the typical Greek numerical prefixes that you might see elsewhere. The other key thing that we need to indicate with our carbohydrate names is what kinds of functional groups we have. So even though we always have the CnH2ON formula, we will have a little bit of variety in terms of what functional groups we have. So what we'll see is we'll see a lot of alcohols, but then we'll also see some carbonyl functional groups as well. And of course, between our carbonyl functional groups, we have aldehydes and ketones, and these will be indicated in our sugars as either aldoses, sugars containing aldehydes, or ketoses, sugars containing ketones. And all of our sugars will contain alcohols. So with a couple simple naming rules, we can indicate the size of our sugars and the different function groups that might be present. And again, this is mostly what we need for our simple sugar monomers because they're all generally sharing that same basic formula of CnH2ON. One of the interesting things about our sugars is that we see a lot of different isomers with the same formula. Again, when your basic formula is CnH2ON, you don't have a lot of variety in terms of your actual formula. So you have to get that extra variety through creating a variety of isomers. So these can be constitutional isomers, the most common of which would be a changing between our aldehyde and ketone. So these are both constitutional isomers of each other, where we've just switched up which carbon has our double bond, whether it's a ketone in the middle of our sugar or an aldehyde with that carbonyl at the end of our sugar. So these would be two examples of an aldotriose and a ketotriose, both with the same formula but different functional groups. In addition to our constitutional isomers, we'll also see a lot of stereoisomers with our sugar molecules. So it's very important that we draw our monosaccharides, our individual sugar monomers, in order to represent that chirality and our different stereoisomers of our sugars. So the common way of drawing sugars is using Fisher projection formulas. So remember, the key with a Fisher projection is that our horizontal arms are always pointing out towards us on wedges. So you can think of them as coming out to try to hug you. So these are the hugging arms of our sugars. And then the vertical bonds are going away from you. So this is the 3D representation of our Fisher projection. But in our actual Fisher projection, these will just all be shown as straight lines. Right? So when you see it written out like this is almost always representing horizontal bonds towards you and vertical bonds going back away from you. So there are a wide variety of different stereoisomers that we see for our carbohydrates. Right? For each of our carbohydrates, we have generally carbons with alcohols and then a carbon with a carbonyl. Every one of these alcohol carbons in the middle of our sugar are chiral centers, right? If you look at them, they each have 4 unique groups: the alcohol, the hydrogen, the group that has our carbonyl, and the group that doesn't have our carbonyl. So we have multiple different chiral centers along our structure here, which then create many different stereoisomers for our sugars. Now, as we've seen before in organic chemistry, right, enantiomers are specifically stereoisomers that are non-superimposable mirror images. Now, as soon as we have enantiomers, we need to designate the configuration of each of our chiral centers so that we know what our stereoisomer configuration is. So normally for our sugars, we're going to include a D or an L to indicate the stereoisomer of our sugar. D and L are commonly used in biochemistry to indicate our chiral centers, as opposed to the R and S notation that we saw in organic chemistry. So D would mean we rotate to the right when we're determining our priority of our groups at the chiral center, and L would mean we're rotating to the left or counterclockwise. In the case of our Fisher projection, as long as we have our carbonyl on top and our carbons lined up below it, if it's on the right side, it's going to be in the D configuration. And if it's on the left side, it's going to be in the L configuration. That referring to the hydroxyl group of our carbon chiral center specifically. So this carbon here is what we use in order to designate this as D-glucose, which of course is one of our aldohexoses. Notice that only the final carbon is actually used here within our name, right? So that D is just referring to this final chiral center. The other three chiral centers are actually implied by the glucose name. So as we change up these chiral centers, we'll change up the name of our sugar. So glucose is a common name that indicates an aldohexose with this particular arrangement of our chiral centers here. So if this is D-glucose, what would the structure of L-glucose be? Well, again, that letter there refers to our final chiral center. So it might be easy to imagine that all we have to do is switch our chiral center at the end over to the left, and we would have L-glucose. In fact, this is actually L-idose. So all of our sugars are named such that the L and D form are always exact mirror images or enantiomers. So our L-glucose would actually have every single chiral center here flipped. So anywhere where our OH is on the right, it would now be on the left. And we could just draw our complete mirror image based on our original D-glucose structure. So L-idose just switches that last chiral center, whereas L-glucose would switch every single chiral center as we're building that structure there. So it would look like this in terms of our chiral centers. Now remember with our enantiomers, so our enantiomers are always perfect mirror images of each other that cannot be superimposed, right? So no matter how you rotate them, they never look exactly the same, but they are exactly the same in terms of what molecules they have, right? So it's purely a shape difference and not actually a difference in terms of the overall atomic composition there. So our D and L isomers for any given sugar will have exactly the same structure, but mirrored. And so they'll also have very similar physical properties, right? So D and L glucose both have the same water solubility, but they actually behave very differently in the body, right? Because they have different shapes that would change how they interact with other biological molecules. In the case of glucose, this actually means that L-glucose can't be used by the body as a fuel source, while D-glucose can. So L-glucose was actually proposed as a diet sugar at one point in time. It ended up being too expensive to synthesize, but it is effective as a sweetener without actually providing that extra calorie intake. One of the reasons why L-glucose was so expensive and failed as a diet sweetener is because most of our hexoses in living organisms are found as the D stereoisomers, right? So just as we saw with amino acids, our biological systems don't create equal amounts of each of these enantiomers. So there are a few sugars that are found in the L form, such as L-arabinose, but we'll most commonly see D form sugars, such as our D-glucose, which is our common nutrient in the body. So as we saw with L-idose and D-glucose, most of our different sugar stereoisomers will not actually be enantiomers. So we would refer to these as diastereomers. So diastereomers refer to any stereoisomers that are not mirror images. We'll actually see much more diastereomers compared to enantiomers within our sugar monosaccharides. Unlike our enantiomers, our diastereomers will have different physical properties. So we have a different relative positioning of our different functional groups, which will actually change how it interacts and then how its physical properties then are. So for example, here we have D-erythrose compared to D-threose. Notice how we change the configuration at our first chiral center while keeping the second chiral center in that D form for both of these sugars. And our water solubilities are actually different for each of these two isomers. Our D-erythrose and D-threose diastereomers here are actually specifically referred to as epimers. So epimers is a subclass of diastereomers that differ at only one chiral center. So we'd say that D-erythrose and D-threose are diastereomers and they are epimers. They are not, of course, enantiomers because they're not perfect mirror images of each other. There are many different important epimers within our body. For example, D-mannose and D-galactose are both epimers of D-glucose. D-mannose differs here at the second carbon, our first chiral center. Notice that we've switched from the right to the left between our glucose and our mannose. And D-galactose switches at the fourth carbon, our third chiral center, switching again from the right to the left. You might remember, of course, the L-idose that I showed you previously is also an epimer of glucose. In that case, we switched the final chiral center, which then switched us from the D form to the L form for our sugar overall. Notice that as we change each of these chiral centers that aren't the final chiral center, we always end up changing the name of our sugar as well, right? So glucose, again, specifically refers to this arrangement of our chiral centers. Mannose and galactose refer to these altered arrangements of our chiral centers. Note, while D-glucose and D-mannose are epimers here, as well as glucose and galactose, galactose and mannose are not epimers. They are just diastereomers, right? Because they differ at both the second carbon and the fourth carbon. Now that we've seen the basics of our monosaccharide structure, I want to look at some of the common monosaccharides that we see in our body. Ribose is, of course, our standard 5-carbon sugar. It's also found in our DNA and RNA. Glucose is our standard 6-carbon sugar. It acts as a key energy source throughout the body. Galactose and mannose are both epimers of glucose that are also commonly used as energy and are used in many different polysaccharides as well. Fructose is the ketose form of glucose, so it's actually a constitutional isomer of glucose, and it's also used as an energy source. These 5 sugars are the most important sugars for us to know in biochemistry. However, we'll take a brief look at our other sugar structures, just so you can see the different variety of sugar molecules as well as isomers of sugar molecules that could be formed. First, we'll look at our D-aldoses. So our aldoses, of course, will always have the aldehyde at the end as our carbonyl functional group, and then we'll have a number of different carbons with alcohol functional groups depending on the size of our sugar. For our 3-carbon triose, we only have one possibility of D-glyceraldehyde. Since each of our carbons has to have either an alcohol or that carbonyl in order to fulfill our CnH2ON formula, there aren't really any other possibilities here. We could put the OH on the left to form L-glyceraldehyde, but of course, we're mostly concerned with our D-aldoses, since those are the ones that are commonly found in our biological systems. When we move up to 4 carbons, we'll have 2 different possibilities, right? So again, for our D sugars, the last chiral center will always be on the right, but now we can put our second chiral center and our chiral center at the second carbon, rather, on the right or the left to create 2 different sugars. Note how our name changes based on our chiral configuration. D-erythrose is the more common sugar here. We'll talk about that a little bit later in biochemistry, and D-threose is much less common. When we move up to 5 carbons, now we have 3 chiral centers here. With those 3 chiral centers, that increases the number of possible stereoisomers we have. So normally the number of stereoisomers would be determined by 2 raised to the number of chiral centers, right? So 2 to the third would be 8. In this case, we only have 4 structures because the other 4 would be L sugars, which we aren't concerned about at this time. So here we have D-ribose. So D-ribose has all of our sugars in the R or D form, all of our chiral centers. This, again, is our most important 5-carbon aldose. D-arabinose and D-xylose are also somewhat common. They, of course, will switch our configurations at the 2nd and the 3rd carbon, respectively. So these would both be epimers of ribose. And finally, we have D-lactose over here as our relatively uncommon sugar. And finally, let's look at our D-aldohexoses. So here we have 4 chiral centers with each of our carbons in the middle acting as a chiral center. So that gives us 2 to the 4th different possibilities, or 16. But then again, we divide that in half as we're looking at only our D sugars here. So our key sugars are noted here as D-glucose, D-mannose, and D-galactose. We also have a variety of other sugars here. Notice here's our D-idose, the D form of the L-idose that I showed you previously. D-allose is a nice sugar because it has all of the alcohols on the right side, so it's very easy configuration to remember, right? It's allose because they're all in the same configuration. Again, you don't have to worry about remembering each of these sugars. We just want to focus on those more important sugars that we commonly see in biochemistry. Now let's take a quick look at our ketoses. So our ketoses will actually have less possibilities for stereoisomers compared to our aldoses. And that's because as soon as we put our ketone in the middle of our sugar, it actually reduces the number of chiral centers that we have, right? Our carbon at the top is not a chiral center because it has 2 hydrogens. Our carbon at the bottom is never a chiral center because it also always has 2 hydrogens. And so we only have our 1 internal chiral center for our 4-carbon ketose. Our 3-carbon ketose actually has 0 chiral centers, and so we only have our 1 achiral dihydroxyacetone as our ketotriose. For our 4-carbon ketoses, so we have 2 forms again, a D-erythrulose and an L-erythrulose. Notice the similarities in name here between our ketose and our aldose. For our aldose, we had D-erythrose, and for our ketose, we have D-erythrulose. So this will be a common naming pattern that we'll see, as we commonly see this ULOSE ending to indicate a sugar that is very similar to an aldose, but it's the ketose form. As we move to our 5 and 6 carbon ketoses, so again here note the reduced number of possibilities due to the elimination of a chiral center by our ketone. So with 5 carbons, we only have 2 chiral centers and 4 different possibilities, 2 of which are our D ketoses. And with our 6 carbons, we only have 3 chiral centers. And again, that gives us our 8 possibilities with 4 D forms of our sugars. Notice we have, again, D-ribulose as the ketose form of ribose, and D-xylulose as the ketose form of xylose. Unfortunately, our glucose breaks this rule, as we have D-fructose as the ketose form of glucose. Again, D-fructose is one of the key sugars in our body, and so you should try to learn this structure. So in this lecture, we got a quick overview of the different varieties of sugar monosaccharides. Again, we don't have as much variety here in terms of our functional groups, but we do have a lot of variety in terms of the different stereoisomers that we can make just by changing around the position of those various hydroxyl groups. Now, even with this relatively low variety of our sugar molecules, there are still a lot of interesting things that we can do with our sugars. There are different ways that we can modify them, and there are also some key reactions that we can have with just our alcohols and our carbonyls. So we'll be looking at those in the next few lectures.